Imagine your immune system as an elite security force.
Right.
These cells are highly trained, they patrol your body's borders,
Yeah.
and they're generally ruthlessly efficient at taking out threats.
Usually, yeah.
But what happens when a bud tumor doesn't just hide from this force, but actually actively corrupts its newest recruits?
That's the terrifying part.
Welcome to this deep dive. Today, we are unpacking a phenomenal 2026 study. This was published in Cancer Cell by Bowie and colleagues, and the data set here is just wild.
Oh, it's massive.
Yeah, it's a massive single-cell transcriptomic atlas crossing these really diverse human clinical cohorts, plus a highly accurate rat model.
Yeah.
So our mission for you today is to solve a core mystery of breast cancer.
A huge mystery, really.
Right, which is why do some early contained breast lesions, ductal carcinoma in situ, or DCIS, why do they stay put while others just aggressively break out and invade the surrounding tissue?
It is arguably the million-dollar question in modern oncology. I mean, for decades, the field mostly looked at the cancer cells themselves for the answer.
Searching for, like, a master genetic mutation.
Exactly. Searching for some mutation that flips the switch to invasion. But this source material shows that the answer isn't entirely inside the tumor's genetics.
Okay.
It actually relies on a bottleneck called immune escape. To survive that transition from DCIS to invasive breast carcinoma, or IBC, the tumor essentially has to build a corrupt environment.
Like it's building a safe haven.
Right. And the star player in this cellular heist is a previously unrecognized, highly proliferative immune cell. They call it the cycTreg.
Which stands for cycling regulatory T cell.
Spot on.
Okay, so we're moving from the overarching concept of breast cancer progression right down to the specific cellular cast of characters.
We have to meet the suspects to understand the crime.
Love that. And regulatory T cells, or Tregs, are usually the essential brakes of our immune system, right?
Yes, they suppress immune responses to prevent our bodies from attacking our own healthy tissues. But here, the tumor is hijacking that braking system. So if a mature Treg is the traffic cop stopping a localized immune response, what exactly separates a normal Treg from this newly discovered cycTreg?
So to map that out, the researchers ran single-cell RNA sequencing across 72 human patients.
Yeah, 72. That's a lot of single-cell data.
It's a mountain of data. It gave them high-definition resolution of normal breast tissue, pre-invasive DCIS, and fully invasive IBC.
Okay.
And they isolated this cycTreg state. Biologically, it is fundamentally immature, yet incredibly active.
Immature but active.
Yeah. The profile has 3 key markers. First, it has intermediate levels of FOXP3.
Wait, let me pause you there. Intermediate FOXP3, because I always thought FOXP3 was like the definitive on switch for Tregs.
It is the master regulator gene, yes.
Right, so if the expression is only intermediate, are they even fully functional suppressors yet?
That's exactly the right question to ask, because no, they aren't fully mature. Their suppressive machinery is still kind of coming online.
Yeah. And the second marker, they have intermediate levels of the transcription factors, IKZF2 and IKZF4.
Transcription factors being the proteins that read the DNA and tell the cell what to build.
Exactly. But the third marker is what truly defines them. They express extremely high levels of MKI67.
Ah, okay. And MKI67 is the classic biological flag for active cellular proliferation.
It is?
So these cells are just trapped in a state of rapid division.
Exactly. And the timeline of when these cycTregs appear is staggering. If you look at a normal human breast, cycTregs are virtually absent.
Not there at all.
Right. The immune environment is mostly dominated by tissue-resident CD8 positive killer T cells.
Good guys. The SWAT team.
The SWAT team, exactly. But in early contained DCIS, these cycTregs start showing up at low levels.
Like scouts.
And then in invasive IBC, their population absolutely explodes. In some invasive tumors, these cycTregs expand to make up anywhere from 4% to 55% of all the tumor-infiltrating lymphocytes.
55%. That is a massive takeover.
It's a complete hostile takeover of the local immune system.
So to help conceptualize this for you listening, I've been thinking of an acronym to trigger the memory. Think of CYC, constantly yielding copies.
Oh, I like that. Constantly yielding copies.
Right. Because they aren't fully mature breaks yet. They are immature cadets, actively multiplying on an assembly line, positioned exactly at that evolutionary junction between contained DCIS and invasive cancer.
That is a highly accurate way to visualize it. They are precursor cells providing the tumor with a continuous self-renewing supply of suppressive cells.
A literal factory.
Yes, and this overwhelming wave of Tregs essentially exhausts and shuts down those CD8 positive killer T cells trying to clear the lesion.
Okay, so let's do a quick recap for you guys listening, just to bullet point this first subtopic.
Great idea.
Bullet 1 normal breasts have tissue-resident CD8 positive killer cells. Bullet 2 in DCIS, cycTregs begin to appear.
Marked by that MKI67 proliferation flag and intermediate FOXP3.
Right. And bullet 3 in invasive cancer, there's a massive expansion of these cycTregs serving as precursors to mature suppressors.
Perfect summary.
So here's a question for you, the listener, to mull over. Think about your own field of work. Have you ever noticed a transitional phase where a small, seemingly harmless anomaly suddenly multiplies and just changes the whole system?
That's exactly what cycTregs are doing here.
Right. But, biological systems don't just infinitely multiply in a vacuum.
No, they don't.
If these cycTregs are proliferating at such an alarming rate, something has to be flipping the switch. Who is instructing these cadets to multiply?
Right. Cells require specific external signals to enter the cell cycle. And the researchers track this back to a specific type of antigen-presenting cell.
The intelligence officers of the immune system.
Exactly. They identified a subset called the type 2 conventional dendritic cell, or cDC2.
Okay, cDC2.
Yeah, and it's marked by a specific protein called CLEC10A, using spatial transcriptomics.
Which is basically a way to map where exactly in the tissue these genes are turning on, right?
Precisely. They mapped the physical architecture of human tumors, especially highly aggressive triple-negative breast cancers.
And what did they find?
They found that these cDC2s are in direct physical contact with the cycTregs.
Wait, so they are actually touching. It's not just some chemical signal floating through the tissue. It's a physical interaction.
A literal physical embrace. They form what we call an immune synapse.
How does that synapse actually drive the Treg division, though?
So the cDC2 utilizes HLA class 2.
Which you can think of as the presentation tray holding the antigen, right?
Good analogy. So it uses that tray along with these co-stimulatory molecules called CD80 and CD86.
Okay.
And these molecules physically handshake with receptors on the cycTreg, specifically the receptors CD28 and CTLA4.
Okay, so we have a very specific biochemical handshake, HLA class 2 and CD8086 on the instructor cell, reaching out to CD28 and CTLA4 on the cadet cell.
Exactly.
But, correlation in tissue doesn't always equal causation. Just because they're touching doesn't mean it's causing the division. How do we know this specific handshake is what's actively forcing the cycTregs to multiply?
They proved causality with a brilliant ex vivo co-culture experiment.
Oh, tell me about this.
The scientists extracted T cells from a tumor-draining lymph node and placed them in a dish with cDC2s taken directly from a tumor.
Okay.
And to track the division, they used CFSC dye dilution.
Okay, so for those who might not work with it, CFSC is fascinating. You load the parent cell with this fluorescent dye.
Right.
And when that cell divides, the dye is split perfectly evenly between the two daughter cells, meaning half the fluorescent.
Exactly. Every generation gets dimmer. And when they paired the T cells with those tumor-derived cDC2s, the T cells rapidly multiplied.
How rapidly?
They diluted the dye so much that they reach what is classified as division 4+.
Wow. Meaning they have that dye 4 separate times.
It is explosive, exponential growth. But, and here is the critical mechanistic proof, if they chemically block that HLA class 2 handshake, the multiplying stopped.
In its track.
Completely stopped.
So the physical connection is absolutely mandatory.
Yes.
But does any cDC2 do this? Like if I take a cDC2 from completely healthy tissue, will it force the T cells to divide like that?
No. And that is maybe the most insidious part of this mechanism.
Really?
Yeah, when the researchers ran the exact same experiment using cDC2s extracted from early microscopic lesions rather than a full microscopic tumor, the T cells hardly divided at all.
Wow. So the tumor microenvironment actually brainwashes the cDC2.
It absolutely does.
The dendritic cell has to physically reside inside the mature tumor, soak up that corrupt localized environment, and alter its own programming before it gains the superpower to teach the cycTreg cadets to multiply.
The tumor conditions the instructor, and the instructor forces the cadets to divide. It's a highly localized feedback loop of corruption.
That's terrifying. Okay, time for another quick bulleted recap for you.
Let's do it.
Bullet 1 cycTregs don't expand on their own. They're driven by direct physical contact with tumor-localized cDC2 cells.
Marked by CLEC10A?
Yep. Bullet 2, the interaction relies on the HLA class 2 and CD80 to CD28 and CTLA4 immune synapse, that physical handshake.
Right.
And bullet 3, only cDC2s, specifically conditioned by the macroscopic tumor, have the power to force this rapid division.
Perfect.
So listener, a question for you. If you were designing a drug right now, would you target the cycTreg itself, or would you go after the cDC2 instructor handing out the orders?
It's a tough choice, therapeutically speaking.
Right. But moving on to dividing cells need a massive amount of metabolic energy.
They do.
We're talking about duplicating entire genomes, rebuilding cellular machinery, and doing it rapidly. They can't just handshake and multiply forever without a robust fuel source.
No, they'd starve.
Exactly. So where's the structural support coming from?
This is where we really have to look beyond the immune system entirely.
Okay.
Tumors are complex organs. They aren't just a ball of cancer cells. They have a scaffold, a structural environment called the stroma.
The structural foundation.
Yeah, and the researchers zeroed in on a group of cells called cancer-associated fibroblasts, or CFs.
Okay, fibroblasts usually just build connective tissue, right?
Right.
Collagen.
Usually, yes. But here, they isolated inflammatory CFs, specifically ICFs, which are marked by the genes CD34 and IL-6.
So we are bringing the structural building blocks into the equation. How are these fibroblasts interacting with our immune cells?
The researchers utilized a computational mapping tool called CellChat.
CellChat. That sounds like a messaging app for cells.
It basically is. It analyzes receptor-ligand interactions across single-cell data to map cellular communication networks.
That is so cool.
And they discovered that these ICFs act as the primary signal senders in the tumor microenvironment.
And what's their main broadcast?
A specific signaling protein called interleukin-33, or IL-33.
IL-33. That's fascinating because that usually functions as an alarmin, right? Like it's released during cell damage to sound the alarm.
Normally, yes. But here, the structural scaffold is actively, continuously secreting it to feed the corrupted security force.
Wow. And the receiving end.
Highly tuned. The cycTregs heavily express the IL-33 receptor called ST2. It's formerly known as IL-1 or IL-1. Yeah, and what's incredible here is the sheer specificity of this survival loop. Picture ST2 as a specialized satellite dish mounted exclusively on the cycTreg.
I love that visual.
Right. And IL-33 is a continuous broadcast of survival rations beamed out by the ICFs, meant only for that satellite dish.
The CCAFs are broadcasting the survival codes, and only the cycTregs have the exact dish tuned to that frequency to receive them.
Exactly.
But how close do they have to be? I mean, is this a systemic signal floating through the blood, or is it highly localized?
It's extremely localized. They prove this using IMBI, which stands for multiplexed ion beam imaging.
Which allows you to look at intact tissue at a subcellular resolution, right?
Down to the exact physical distances between cells.
Yeah.
And when they map the human tissue, the physical proximity between the CHEFs, the cycTregs, and the DCIS epithelial cancer cells strongly correlated with invasive progression.
So they are huddled together.
Yes. Furthermore, when the cycTregs receive that IL-33 fuel, it not only promotes their survival, but it heavily drives their anti-inflammatory polarization.
Meaning it makes them even better suppressors.
Exactly. It triggers them to release more IL-10, which acts like a wet blanket on any nearby CD8 positive killer cells.
That is, honestly, it's a terrifyingly elegant ecosystem.
It really is.
Imagine finding out your own structural cells, the scaffolding of your breast tissue, are actively colluding with hijacked dendritic cells to build an army of immune suppressors.
It's a full-scale betrayal.
Let's recap this stroma subtopic for the listeners. Bullet 1, the tumor structural cells, the CAFs, actively keep the cycTregs alive.
Specifically the inflammatory ICFs.
Right. Bullet 2, these ICFs secrete IL-33. And bullet 3, the cycTregs receive that IL-33 via their ST2 satellite dish, promoting their survival and increasing IL-10 to suppress the good guys.
Spot on.
So listener, does it surprise you how orchestrated this communication is between completely different cell types? We've got epithelial, myeloid, stromal, and lymphoid cells all coordinating to raise a tumor.
It takes a village, apparently.
A very corrupt village. But understanding this biology leads us to a critical pivot. What does this all mean for a patient sitting in a clinic today with a DCIS diagnosis.
That is where this paper transitions from incredible biology to potentially practice-changing clinical diagnostics.
Okay, let's hear the clinical data.
The researchers interrogated massive human clinical databases. Specifically, they looked at the HTN DCIS cohort, as well as pure DCIS and IBC cohorts from Seoul National University, or SNU.
The SNU cohorts, right?
And they discovered a profound statistical correlation. Earlier, we noted that cycTregs are very rare in early pure DCIS.
Right, making up less than 1% of the cellular landscape. They're just starting to appear on the assembly line.
Exactly. But despite being incredibly rare, a high relative abundance of cycTregs in a primary DCIS lesion predicts a significantly shorter time to recurrence.
How significant?
The hazard ratio they calculated was 3.321.
Wait, I have to push back on the math there.
Go for it.
A hazard ratio of 3.321 means patients with high cycTreg levels are over 3.3 times more likely to see their cancer return or progress.
Yes.
But if these cells make up less than 1% of the total cells in the lesion, how can they wield such massive predictive power? Are they actively causing the recurrence, or are they just a byproduct, like a canary in the coal mine?
It's a crucial distinction. They aren't just the canary, they are the miners laying the dynamite.
Oh, wow.
Yeah, because they are precursors, right? Their presence indicates that the tumor has successfully established the immunosuppressive assembly line we just talked about.
The cDC2s and the ICA-HEFs are in place.
Exactly. Even if the factory has only produced a few cycTregs so far, the fact that the factory is fully operational guarantees future suppression. It means the immune escape bottleneck has been breached.
So it's not about the current volume of the cells, it's about the biological trajectory they represent.
Exactly. And this bears out in later stage disease, too.
What happened there?
When they analyzed the metabric cohort, which focuses on invasive breast cancer, the presence of cycTregs strongly predicted poor disease-specific survival.
Meaning higher cycTregs equal a higher chance of dying from the disease.
Yes. High cycTreg levels perfectly correlated inversely with CD8-positive T cells. More cycTregs meant fewer good guys and ultimately worse outcomes for the patient.
Makes sense.
And here's the kicker. In grade 1 DCIS,
which is the least aggressive-looking form under a microscope, right?
Right. In grade 1 DCIS, the cycTreg score was the only immune subset associated with recurrence.
Wait, really? The only one?
The only one.
Okay, quick recap for the diagnostics. Bullet 1 high cycTreg levels in DCIS predict a 3 times higher risk of recurrence.
HR of 3.321.
Yep. Bullet 2 in IBC, cycTreg presence predicts poor survival. And bullet 3 high cycTregs perfectly correlate with low CD8-positive killer cells.
Exactly.
So listener, if a simple biopsy could give you your cycTreg score early on, how might that change the aggressiveness of your treatment plan?
It could completely alter it. It gives oncologists a window into the tumor's future behavior.
But diagnostics are only half the battle, right? We know how they grow, we know who fuels them, we know they are deadly. How do we break the ecosystem? How do we destroy them?
To test therapeutics, the researchers utilized a highly specific animal model, the outbred, NMU-induced Bragdoli rat model.
Okay, why rats? Why not the standard, inbred lab mice everyone uses?
Because standard lab mice don't always reflect human complexities. This specific outbred rat model much more accurately mimics the genetic diversity and the precise immune-evasive mechanisms seen in human breast cancer.
Ah, okay. Makes sense.
So using this model, they deployed 2 distinct therapeutic strategies. Therapy 1 targeted a receptor called OX40.
OX40. Okay, so we're going after the cell's surface receptors.
Yes. The gene for this receptor, TNFR-SF4, was identified as a major hub gene on cycTregs.
A hub gene meaning it's highly connected and central to the cell's identity.
Exactly. In fact, 74% of all cycTregs strongly express OX40. So the researchers engineered a dual therapy algorithm. They combined an agonistic OX40 antibody with an anti-PD-L1 immune checkpoint inhibitor.
And to prove this actually stopped the proliferation, they didn't just measure the tumor size, right? Because that can be imprecise.
No, they tracked the actual cellular life cycles.
How?
They used an EDU and BRDU dual pulse chase experiment.
Okay, you're going to have to explain that one.
It's a highly precise kinetic tracking method. EDU and BRDU are both thymidine analogs.
Meaning they look like the letter T in the DNA sequence.
Right. And when a cell replicates its genome during the S phase of division, it incorporates these analogs directly into its new DNA.
So if you pulse the system with EDU, wait a specific amount of time, and then pulse it with BRDU, you can see exactly which cells were dividing at what specific time point based on which analog they incorporated.
Precisely. It allows you to track the exact kinetics of the population over time.
That's incredibly clever.
And the tracking proved that the OX40 and PD-L1 dual therapy actively and specifically suppressed the cycling kinetics of the cycTregs.
So the proliferation plummeted.
It plummeted, and by removing that constant wave of suppressive cadets, the CD8 positive killer T cells were finally able to flood back into the tumor.
So targeting OX40 basically triggers a self-destruct or halt production code on the assembly line.
Exactly.
But what about therapy 2? You mentioned the tumor stroma earlier.
Right. Therapy 2 went after the environmental fuel line. They neutralized IL-33.
The survival broadcast from the CYCFs.
Exactly. They administered an anti-IL-33 antibody to the rats. By blocking those survival rations, the cycTregs were essentially starved out of the microenvironment.
Cutting off the supply lines.
Yeah.
And how did the broader immune system react to that starvation tactic?
The results were profound. Not only did the primary tumor shrink, but there's a massive systemic explosion of memory effector CD8 positive T cells.
Like experienced SWAT team members.
Yes. These are the highly lethal killer cells marked by the EOMES protein, EOMES. And this explosion didn't just happen inside the tumor bed.
Where else did it happen?
They observed it systemically, all the way out to the bone marrow and the thymus. By neutralizing IL-33, they didn't just weaken the tumor, they reawoke the entire systemic immune response.
It's incredible. Time for our final recap. Bullet 1 dual therapy with an OX40 agonist and anti-PD-L1 directly halts cycTreg proliferation.
Verified by that EduBU tracking.
Right. Bullet 2 neutralizing IL-33 cuts off the CAV support loop and starves the cycTregs.
Yes.
And Bullet 3, both treatments shift the tumor microenvironment from immune-suppressed to immune-reactive, drastically increasing those EOMES positive effector CD8 cells.
Perfectly summarized.
So, listener, a question to think about. Between hacking the cell's internal receptors with OX40 or cutting off its environmental fuel by blocking IL-33, which strategy do you think holds more promise for complex, evolving systems like human cancer?
Realistically, clinical application will likely require a combination of both strategies tailored to the individual patient's specific tumor ecosystem.
Right, because cancer is an ecosystem. That's really the grand synthesis of this entire data set, isn't it?
It is. The transition from a pre-invasive DCIS lesion to invasive breast cancer isn't just a random genetic accident.
It's a highly orchestrated multicellular immune escape.
Exactly. Driven by cDC2 instructors directly educating cycTreg cadets, heavily supported by an exclusive IL-33 supply line secreted by inflammatory fibroblasts.
And by mapping every physical interaction and signaling loop of this ecosystem, science has basically handed us a powerful new prognostic biomarker in the cycTreg score.
And equally important, it has identified targetable vulnerabilities in OX40 and the ST2 to IL-33 axis.
This is the kind of biology that genuinely has the potential to revolutionize how we manage breast cancer care.
It absolutely does. But, as we wrap up, I want to leave you with a final, slightly unsettling biological implication. Yeah. We've established that inflammatory fibroblasts and cDC2s are building these incredibly complex communication networks to nurture cycTregs before the tumor even becomes invasive.
Right.
So it raises a fascinating question. Are there microscopic, undetected conversational shifts happening between cells in completely normal-looking tissues right now, just waiting for a secondary biological trigger to start the assembly line?
Wow. If the immune system is our elite security force, it really makes you wonder if the corruption starts with a single localized whisper long before the alarm ever sounds.
Something to think about.
It highlights just how vigilant our biological surveillance needs to be. Thank you for joining us on this deep dive into the Bouillet-Ellis single-cell atlas. Keep questioning the microscopic worlds within, and we'll see you next time.